What a pleasure, Joana. It's been too long. Joana and I go back a long way, at least 3 children.
Plus one, plus Lida.
Plus Lida. This is exactly what I was going to talk about. We, through all these wonderful things we've been doing over the years, come up with all these beautiful ideas. And as you know, I'm a doula. I'm somebody who likes to help people bring things into the world. And I think Lida certainly has become a beautiful, beautiful kid. But I think you're going to show us something even more exciting today, which again, I'm always excited to see all these new ideas come around. I know you've already spoken to many people in the room, and there's a sense of general kind of excitement, I think, and with good reason, because of course, some of this work you've just published in Nature Communications, I think it was a beautiful paper. I'm hoping you will tell us about that today, amongst many other things, I'm sure. So really, all that remains for me is to thank you for coming. I'm sure we'll have a great seminar, and then afterwards we'll do the usual thing, get more ideas. Thank you.
Thank you. Thank you, Martin, for everything. For me, it's really a pleasure to be here. And Martin is a man of my life. I have different types of men of my life. And because he really supported, and it's someone who always kept me motivated, and has this saying, like, I'm always looking for new things and exciting things to keep doing research. So for me, it's a pleasure to be here and to discuss, show you my latest insights in trying to understand brain function. I've always been very intrigued by oscillations, so I hope you'll understand what I mean by that, and also share some of the excitement that I will go through. So today, the title of my talk is Oscillatory Modes at the Basis of a Brain Melody in Health and Disease. This was also in collaboration with Martin, the part of the melody. So overall, why is it not going forward? Okay. So this is just an outline of my talk. I will focus a bit on the motivation, why I'm doing this, what are these patterns of activity that we observe at the macro scale. Today I will focus in fMRI signals, and then I will show results that are supporting this hypothesis that has been put forward by many even before me, of the role of this kind of resonant waves that may be at the origin of functional networks. And then I will also discuss some points showing you new insights that I'm getting that are still unpublished. So overall, I'm an engineer, and I'm very curious, and I think I took the challenge in my life to dedicate it to try to understand how the brain works. And that's what I understand as brain function, right? And our best correlate of brain function is this nearly infinity of activity patterns that we detect across modalities that relate with cognition, emotion, or behavior. But then when function is not working, that is the thing, we have problems of dementia, psychiatric disorders, disorders of consciousness. If we look at the brain as a system, something is going wrong. And this is the real problem, let's say. It's the problem that we, beyond like the hard problem or soft problems of consciousness or everything, there are really millions and millions of people suffering with disorders. We all know someone who suddenly starts speaking nonsense. There are very little solutions. And the thing is that we still don't understand what is brain function, so we cannot treat it. And because we don't still gather what are the fundamental principles, the mechanisms, or in physics they call what are the constitutive equations that support brain function, right? And so we can look at the structure. And here I look at the brain structure as not only the neurons and the neurons connected together, but considering also, I think it's important to take into account that the brain is a complex system, not just a complex network, but also has membranes and fluids and molecules and ions, and there are different ingredients. And maybe overall, if we can consider these things or understand which are the key ingredients, we can build mechanistic models, right, to try to understand this. And even beyond just the brain structure alone, it's important now more and more people are considering also how the whole body interacts with the brain. So we have the endocrine system that releases hormones that go to the body. I will not go so much on this level, but it's really to understand how different things interact together to ultimately come up to function. And in my research, I look at the macro scale. And this is important, especially when I'm talking to people from pure neuroscience, where I work with many in Portugal as well, who are really just looking at the micro scale. I'm not saying that the micro scale is not important, it is, but it is at the macro scale that we are seeing already, like for 100 years since the first EEG recordings and things, that we detect collective phenomena. So even the rhythms we detect with EEG, it's built up by millions of neurons interacting together, organizing together to generate collective rhythms. And what we see also in fMRI is also a macro scale activity, in the sense that we see really long range interactions. So there are billions of neurons that are working together to form some processes. And we know that this is related to brain function because it is altered in many neurological and psychiatric disorders. Although the one mostly used in clinics is still EEG, fMRI is still not fully used. In the last decades, a lot of research has shown that fMRI patterns of activity are distorted or are altered in psychiatric disorders and neurological disorders. So there's something there at the macro scale that is very important, and at the level of activity. So it's macro scale and activity, so things that change in time. It's important. So when we look at these patterns of activity that we detect with fMRI, one of the striking thing that I think now there's really a consensus that I think hundreds or maybe thousands of studies have found is that there are these macro scale patterns of activity where different parts of the brain exhibit correlated activity, right? And this is assumed, like if you have, for example, a signal in two brain areas, that from time to time you'd see an increase of the signal in some areas and in another as well. And since they are co-activating, we assume that there's a functional connection between these two units. And this is generally understood because, like, okay, let's assume then there's a structural connection, right? So I put there some white arrows just to mimic the white matter connectivity or something that, okay, if the signal increases in one, then it will propagate to the others and it will make the other increase and the others as well. So the areas that are more coupled together, they will correlate together. And this has been like an assumption that then I will discuss on this later. But overall, there are more and more, there's more and more evidence, and now it is really coming through at the psychiatry level. Papers in Lancet Psychiatry, this is quite influential in the sense that they identify like network patterns that are related to different emotions or feelings of people, or even cognitive control. So there are different patterns of activity that are associated with brain function. And these are detected consistently in human fMRI studies, of course, with some variations. We can go through that. They are also detected across mammals. There are these patterns of activity. They are disrupted in disease. They can be modulated by drugs, tasks, transcranial magnetic electric stimulation, by music, as many of you can study that. But their origin remains under debate. Even if we can have some mechanistic models, it's still not fully understood. We still don't know. So there are hypotheses of cluster synchrony, standing wave hypotheses, like Selene has also been putting forward. But then the key thing is, at least challenges me, is to understand what organizes activity at this scale, right? So when we look at the fMRI signals, we don't see the default mode network or these other networks. But the thing is, if we put like regions of interest, okay, we want to see, we take the signal in one, average signal in some regions, average signal in another, and we can see how much they correlate in time, right? So we can build a functional connectivity matrix, which is like a correlation matrix, telling us how much the different parcels, imagining like each line in this matrix is a region of interest in the brain, and we can see how much they are correlated. But this correlation is not static. It's actually dynamic. It means that from time to time, you can see synchronization between these areas. This synchronization can be global, can be local. It can be different modes of patterns of activity can appear. And by looking at this thing, we see there's no stationarity in the sense. It keeps moving all the time. But we also see some kind of stable pattern sometimes. And these patterns reoccur over time. So one way to kind of look at this, I will explore this a bit more, but this suggests that there is a dynamic formation of functional networks from time to time, so it's not a constant process. And so we would see, like, there's some subsystems that synchronize together, then other subsystems, and this is like from one single scan, but if we look at several of them, we see that from time to time we see the DMN taking shape, then the somatomotor network taking shape. So then this is what we get with the LiDAR by just plotting here the leading eigenvector at each instant of time. But to understand this, this captures actually the phase relationships between signals in different areas over time. It's a thing to do. So how do we capture these phase relationships in fMRI signals? If we have just a signal, let's say, so this would be the real signal that we are capturing. If we want to get the phase of the signal, so I tried to make this cartoon, which is like we do the Hilbert transform, which kind of imagines or takes an imaginary component, so it creates an oscillatory signal from the real-time series, and then at each instant of time, I'm plotting the phase, and it's colored according to the angle that it's taking, so we can see and we can plot all the phases in a phase space, so to see the phases of the fMRI signal. So once we do that, we can get one of those little phases at each time point. You can see what is the phase of the fMRI signal in different brain areas. And we can see, for example, here that all the green, the ones that are colored in green, are aligned in phase, and the ones in blue are aligned in phase. And we see some pattern here, so this would evolve over time. Now, when we have all these phases, to characterize them, to characterize the phase relationships, we can take all the little arrows and put them centered together at the same point, right? So we see how all the phases are aligned and organized. And then when we take the leading eigenvector, is as if you would take an arrow that crosses this space, which is there represented, and then this arrow is the one that maximizes how to separate, like how maximize the alignment of all the phases into that vector. So if they could be in anti-phase or in phase with the eigenvector, but this is the vector. And then it allows to split the phase space in two. If everything was red or everything was blue, it means all the phases go together. Everything is going up and down. If there are areas that are with one sign and with the other sign with respect to the eigenvector, it means that when one goes down, the other goes up. It's a bit to have an intuition. It's hard to fully explain how this works, but actually when we do this, when we take the leading eigenvector of each of these matrices, and this was like the one that you could see the video, it evolves over time, right? We can put them all together, we concatenate all the eigenvectors, and then we cluster them to see, are there particular specific types of eigenvectors that reoccur over time? Can we detect some pattern formation there? And so when we cluster all these eigenvectors that are very noisy and everything, and we say, okay, now to the algorithm, separate me these clusters into 5 clusters, and then we look at the centroids, which in reality are like this, are like eigenvectors, and then we color the ones that are shifting in phase with respect to the main orientation. We see the formation of patterns looking like the default mode network, or the frontoparietal, or the somatomotor, or the visual network. So it seems that even over time, there is this formation of these patterns. This allows to interpret brain activity, like the dynamics of the brain, as a trajectory. Imagine we have, for example, here, 7 networks. Each network represents a point in a phase space, which I tried here to illustrate. And the dynamics, so how the brain activity is evolving over time, could be represented as a trajectory in this space. So you may never actually be in one of the attractors, but you move closer to the DMN attractor, closer to the frontoparietal, closer to the somatomotor, and then you can quantify like how much time or how many time points it went to the green zone. And the green zone is the cloud of points that is closer to that centroid with respect to the other. And this allows to, of course, it's an interpretation, but actually it is getting a lot of relevant information. So we've been applying this technique to different data sets, and I just give here an example of different papers that we published, but we already applied this to different data sets. And what is relevant with this type of approach is that we can then look over time, when we have all these time signals from the different brain areas that look very complex, we can identify which are the time points that a given pattern occurred, right? And then we can calculate how often in this scan, how often this pattern occurred. And we count how many blue shades we have, or how often did another pattern. So we can calculate the probability, we can calculate how long they last, that is how many consecutive time points you remain close to the blue centroid, let's say. And also we can calculate switching profiles, which is if you are in the blue centroid, what is the probability of going to the green one or to the other one? So this allows for single scan metrics. So you can resume a scan that has a lot of box, like sometimes it's a gigabyte of data from one scan, and you summarize it into like the probabilities of occurrence of these different patterns. So we call it as well occupancy. I will go a bit on that here, because it's a term that is used from dynamical systems theory. And here we tried, because we understood that, wait, what we're doing is a technique that is already applied a lot of times to characterize complex dynamical systems. Like you see something and you try to see what are the probability of occurrence of this state, and this is called fractional occupancy, which is basically the time points assigned. The dwell time, like I said, is the duration, the mean duration of this state. And then you can also look at the Markov chain transition probability, which is basically a tool used in dynamical systems theory as well, to see, okay, if you are in this state, what is the probability to remain in that state in the next time point, or to switch to state number 2, or to switch to state number 3, and each state there is represented here in the brain by a pattern. Here, instead of representing it as a network, we color the brain areas, but this means, for example, that if we are in this global state, is everything goes together, so the activity in all the brain areas go together in phase. If we are in this state, there's 77% probability, this in the Human Connectome Project data, so more than 100 subjects. So it was 2 scans of 100, 2 sessions of 100 subjects each, and to remain in that state. But then with like 8% probability, you will switch to the default mode network, okay? But then when you're in the DMN, you decay back to the global state. So there is like a state that is more strongly like a baseline state, and then from time to time we switch to these other weakly stable states, and then you return back to them. So this is why in this matrix we see the diagonal has lighter colors, which means that there is a higher probability of remaining there. Even in the DMN, you have 50% chances of the next time point we still detect it, but then it decays down. And then most every time they decay back to state 1, so with higher probability. So you see the arrows. So here it's hard to go up, but then it decays back to 1 with 22%. Here it goes up, and then 26% of the probability of going up. So there are these weakly stable states that return. So this made us like speculate, let's say, because it can give insights on the origin of observed phenomenon, that this would be like a main attractor. And this would be what is called ghost attractors or pseudo attractors, that are solutions where the system tends to, but then when it arrives there, it stays there for a short period of time above randomness, and then it decays back to the baseline state. So this was one of the theories that we've been put forward to try to understand what is going on. But so Lido, I need some water, but it's fine. So Lido was actually, maybe I have, no, I have a bottle here.
Oh,
if you could.
I will fill it up for you.
Predicting that that's still
okay. Time. We will not leave until,
okay, so Lido was developed in the beginning to analyze a data set that we had from 88 healthy older adults, so they were all healthy. But they were all more than 50 years old, and they were characterized as the best and the worst performers from a cohort of 4,000 people. They were selected to go to scanning, so we had half that were very good and half from the very poor group. And the idea was to find, can we detect differences in their activity? And they had tried other things, we kept trying, and we found that if we take these probabilities of occurrence, we could find that, for example, good performers had significantly more of this global mode this baseline. They spend more time in the baseline state, so they have less excursions. And whereas the poor performance had more excursions, especially to these sensory networks, for example. The time was okay, the results are significant, but I don't know how replicable this is. And then we started applying this tool to different types of data sets. Even like in open source data sets, like this one from the COBRE initiative, where they had like 150 people with schizophrenia with respect to controls. We downloaded, we applied the tool, and for example, we see, oh, there's reduced stability of the global mode, like this state that we found that was higher with people with higher cognitive performance, and they have more excursions to other networks and less of the default mode network as well, and more excursions to the visual, to the somatomotor, and even to the limbic. So even when they're at rest, the patients can be characterized by having more erratic excursions to this network. It's interesting, but let's see how much this relates. We also did another, I'm here summarizing a bit the different results, like one on psychiatry. This is to show how a given molecule, a specific compound that enters your blood, passes the blood-brain barrier, does something, attaches to some neurotransmitters, and it specifically modulates one of these patterns. For example, when we give psilocybin compared to all the other conditions, so these are two conditions, let's go slowly. So each subject was scanned 4 times. The first time, before anything was injected, it was scanned. Then either psilocybin or placebo was injected, they were scanned, and then they go back 2 weeks later, and again, they repeat the same procedure without knowing if they were injected with psilocybin or a placebo. And when we compare these 4 conditions, for many of the states, there were no significant differences between the 4 conditions across subjects. So there was some stability in the occupancy of the different networks. Even the DMN at this stage is not showing so much, but there was one particular pattern of activity, so the frontoparietal network that is usually associated with executive control, that was significantly reduced. This was visual. We could really see that there was much less occurrence of this network in all the scans. It went on like to 3% of the time, and compared to all the other conditions, and at the expense of an increase in the global modes. So it supports this hypothesis that, okay, when you remove this attractor, that is not this ghost, you just spend more time in the baseline, but it doesn't affect the others. And this was super nice because it was only found with 9 subjects. I said, no, the stats are really, really strong, so there should be something. We insisted. And now in October, there was a group who replicated this in a completely different, a new data set, also with psilocybin. They also applied LIDA. I didn't know them. And they actually found exactly the same thing, that there was a decrease in the lateral frontoparietal networks. They validated it, and they even added, they changed, instead of using k-means clustering, they used diametrical clustering, so it's more elegant in a way to capture the networks. But I found it, I think it's very nice when you do something and then you find someone else who replicates your findings, because it kind of demonstrates that there's something there. And then, for example, there are patterns of activity. We can make different questions, and here are some examples of studies. You know, the paper maybe from Maria Celeste, where we were seeing which is the network that increases when they are listening to music. And this network actually also correlated with the emotional reward score that people responded to a questionnaire, and it actually showed activation of the orbitofrontal area here of the brain when listening to music, with a collaboration with a group from neurosurgery, where they implanted deep brain stimulation in subjects, and we compare off versus on. So when deep brain stimulation is turned on, there is a highly significant increase of this pattern of activity as well. So it's really, and this is how much it overlaps with reference functional networks. So it's a limbic network that is a very focal limbic network. And then for the paper from Eloise, I thought Eloise would be here as well, but maybe she's watching online. We also found like this network correlated with the happiness score of babies that the women were seeing inside the scanners, right? So the more the children tended to be happy because they had a neutral face when they were seeing in the scanner thing. But it was really nice because it wasn't only just this orbitofrontal network, but also connected to the amygdala and to the hippocampus. So there is, because they had to remember that the children were happier. So it is really returning some sensitive and meaningful information. But the thing is, why? What is at the, at the origin or at driving this formation of these networks? So the idea is to understand, investigate what is going on. And from the perspective of statistical physics, say these intrinsic connectivity networks or functional networks can be approached as this concept of pseudo-attractors, right? But then if there is this term, attractor, the question is, what is the attractive force favoring some pattern configurations with respect to an infinite possibility of patterns? Like, why does activity recurrently returns to these patterns of activity? So one of the things that when we keep looking at the data for a long, long time and you think, wait, they're not telling me the full story. Because when I apply LiDAR, this is in voxel space, we are now doing this in a nicer way. What we find is that there are regions that co-activate together, but when the signal increases in some areas, it also decreases in others. So it creates this kind of pattern of activity that it increases in some and decreases in others. And in between areas of increases of activity and decreases, there are points of no motion. What does this mean? So we can see this periodic co-activation separated by co-deactivations, right? And this is opening the possibility of a fundamentally distinct mechanistic scenario, because this relates, whenever we see this type of things, is when we see like the emergence of a standing wave pattern, right? If we would see, imagine this is a static membrane, and whenever it would just do one vibration, we would see like all the points that belong to all these antinodes will be correlated together, but also in between you will see points where it goes down, right? So you'd have this activity, this was not. So this hypothesis that what we are seeing is actually related to standing waves has been really, as you know, proposed before. Already for EEG, there's a huge literature on that side, but at the level of fMRI, Selena Tassoy was one of the pioneers using, and to prove this, so to test this hypothesis, what people have been trying to do is to go, how can we predict what would be the standing wave? Imagine you have a cord or you have a membrane or whatever, and we want to predict what are the standing waves that can occur from this type of structure. And for that, what Pierre-Simon Laplace proposed was to use the Laplacian, which basically tells you how much the signal diffuses through space, through X, Y, and Z, and you do this for all the points, and when you have that, you identify also the boundary conditions, and then you do the eigenvectors of this Laplacian, and you would take each eigenvector would represent a mode of vibration or a standing wave pattern that this structure could replicate, assuming that it is a diffusive process. This is also important, but in some things. So always there is a fundamental mode, a mode number one that is a global mode. Here in Silen's paper, we see there is this global mode that is like either everything goes red and blue, or here it's the second mode already, because here she's not showing the mode number one, it's already mode number two. But here, for example, there are other papers who are predicting this, and you can predict the Laplacian. Either you can apply it to a graph, so like a network, and you can see which would be the resonant modes in a network structure, and then you apply what is called the graph Laplacian, or other people use a surface-based approach where you take the mesh of the cortex and you can see what would be the modes that this mesh holds. But then there are also since the original Laplacian is defined in 3D space, so you can also predict the modes from a 3D structure directly, right? And this links with music, and you will see how much it does. So the thing is that these approaches, these attempts to predict what would be the modes of vibration or the harmonic modes or the standing waves that the brain structure would permit, you apply this, but then the thing is they also show like what we see, these gradient-like phase distributions. This means that when the signal goes up in the red points, it will go down in the blue points and vice versa. But the match to resting state networks is still not one-to-one. So there is an approximation, but so far it cannot explain exactly, it cannot be proven. So it's still not widely accepted in the field. Because of course you can never account for everything. And the thing is that standing waves are really inherent properties of bounded systems that have some degree of freedom, right? So you have at least some elasticity, or each point in the system has some, they are all coupled together in a membrane, but they are allowed to move. And so you'll always identify a mode where everything's synchronized in phase. It's like the first mode, right, that we see here. But then you can have other modes that can appear on the same system, that, for example, half of the system goes up and half of the system goes down. Now, when you have higher dimensional modes, you can have multiple antinodes, right? So you'll have antinodes that go up together, so they have the same sign, so they will be correlated. And you can have antinodes that go down when the others go up. They will also be correlated, and then they will be anti-correlated between two antinodes with different polarity. And this is observed across fields, across scales, I mean, different things. There's a whole story of this. And then there's also here an important thing that, just for later interpretation, that here what I'm showing is a stable standing wave, right? It's always vibrating, but you come from music as well, and you know that you can, these modes or notes, let's say, that you play, they last for some time, but eventually they decay, right? When you play a note in a piano or a chord, you play it, and then it will, and then eventually it decays. So this is called, it's damped. So it oscillates for a period in time with decaying amplitude. This is usually known, just to go a bit more in this direction, what they are, where they are coming from. So usually we see standing waves, here would be the black line, and it's the result of like a wave, the same frequency, when it interferes. So it's interference of two waves that are going in opposite directions. So it's like it would go, it hits the wall, it comes back, it hits the wall. So you can do this infinite times of times, and the ones that when they come back, they fit in together, right? This will generate a resonance effect, a resonant wave. And this can occur from different perspectives, like in 3D systems, in three-dimensional, imagine these are room modes. Imagine you have a chamber, like these two are the same chamber, and the particles inside this chamber, even this room that we're talking in, if we had just a single tone playing, we could generate these standing waves of activity at the room level. And it doesn't mean that there's necessarily two waves going in opposite directions. Actually, the net displacement is weak. But what is interesting is that if you would put nodes, imagine we had a fMRI scanner or a scanner that could capture the room modes, and we put them in the place of different antinodes, you would see that the antinodes with shared polarity, they are with correlated oscillatory activity, right? And the ones that are in other antinodes, they will have anti-correlated activity with the red between the blue. So this is important because it shows, okay, this is a mechanism that can drive long-range functional or long-range correlations between things in a space, but to prove this and to see if this is really what is driving functional networks, what I was thinking is that we need to check if the functional networks that we are seeing with LiDAR or everything, if they oscillate. Because we see the patterns, they are spatial, but they are static. And I want to see how much they oscillate. But with the temporal resolution that we have usually with fMRI, it's very hard because it's super noisy and you need good temporal resolution to really detect them, because there can be many modes superposing. You never have a room that only has one mode. Probably here there are different modes resonating simultaneously, right? So we had to... So this is where starts now the second part of my talk. To really understand the nature of functional connectivity, I had to kind of disambiguate this or elucidate whether the functional networks or the signal that is correlated across points oscillates or not. So I met Noam Shamesh, that Morten introduced me to him as well, and he is the director of the preclinical MRI lab, and there they have a 9.4 Tesla scanner for rodents. They have something called a cryogenic probe that removes a lot of thermal noise and things, so the signal is very neat. And we asked him, look, I want to see if 2 points from a functional net, if the signal giving rise to the correlations, if it oscillates in time or not. So I need the best temporal resolution. How far can we go? And he said, well, I don't know. We can go like to the scanner and try. And so we went there, trying, you know, like it turns off the alarms to see how much. And we are checking the temperature of the scanner so that it doesn't burn. And we reached like 38 milliseconds of temporal resolution. We tried lower, but then the signal had a lot of artifacts. And here it was really, really neat. You will see. So this means we could have, in a 10-minute scan, we have 16,000 frames. Where sometimes with humans, if we have like a TR of 1, we only have 600. You know, so we really increase a lot. And just to give you, and then we chose, so this is possible if we do a single slice acquisition. Because usually, even when we do our normal fMRI scans in our like humans that we are analyzing, actually the TR for each slice is short. But the thing is that when we want to cover the whole brain, we are doing like, inside 1 second, we do 100 slices. So we're doing like, and this is what also makes a lot of noise in the scanner and everything. If we don't change the slice, and we are only there, first you have much less noise, but you really increase the temporal resolution. I will show you later with the humans. So we chose a slice that is known to show really some of the resting state networks in rats. This is for rats. Just to give you an idea, because it was also for me the first time, like this is the slice with 2 types of staining, but here is the cortex. It's not folded like it is in the humans, so it's easier to really see it. And then here is the striatum, which is like subcortical structures, but just to have it. So, and also I showed these different stainings to show that it can have different textures, right, of the thing. And then our experimental design was very simple. We only did 6 rats, 3 first, and we replicated. And then we just sedated the 6 rats with medetomidine, which is like they don't move, because in animals you cannot tell them now don't move for half an hour. So you need to paralyze them a bit, but at the same time, if you give too much anesthesia, you lose the signal. There's nothing, right? Because there's nothing happening. And medetomidine is used because it really enhances the resting state networks, so you see them a lot, but then it also creates oscillations that people were a bit, I will comment on this, arguing what it was, and they found out that if you add a bit of isoflurane, you kill the oscillations, but you still detect some bold signal. And this is used to do typical task-driven paradigms, like they stimulate the paw of the mouse or the rat, and they see a bold signal activation under this type of thing. And then if you give isoflurane 3%, it's what they call deep anesthesia, and the animals, you can cut them, you can whatever, they won't feel anything, and you also detect anything in brain. And then we also scanned two rats post-mortem, which is important to define a baseline, because you would be amazed by seeing just a dead brain. But if you go into the right resolution, you can see a lot of noise, let's say, but it's just, I don't know, thermal noise or whatever it is. So it's important to define the baseline. So here I will show a video. This is, of course, already the signal has been now band pass filter in a narrow band that I found that had a lot of the signal is very powerful, but I will also show others, just for you to have an idea of the temporal resolution and the scale of the signals that we can see. So when we see here, so I didn't, we clearly see, for example, that there are long-range correlations between opposite sides of the cortex, you see, and then it's periodic, like now it turned red, and then now it turned blue, and then now it turns red, okay? And here, the mask, I didn't do it by hand. I'm just taking only the voxels that have power in this frequency range, and the ones that don't have power in this frequency range above postmortem baseline are transparent, and I will get anatomical image behind. But you see here that we clearly see the cortex. And this was striking because it was quite consistent across rats, but then I will just show here a bit what we did to get to that image. So I decided to analyze the signals, the power, in different frequency bands. So let's say, usually each of these columns here is a different frequency band, up to 0.4 hertz. Okay, these oscillations, for example, 0.1 means that there are oscillations in these voxels that last 10 seconds. Here would be like there are oscillations in these voxels that last 5 seconds or 4 seconds. Here it means that there are oscillations that last 2 seconds. So I could go up and faster and whatever. But so we find that, and here it's the different conditions. So this is sedation, light anesthesia, deep anesthesia, and postmortem. Okay? So we find there's here what I'm showing here is also to show that there's a bit of noise even in the postmortem scans. But what we find is that in all the 3 conditions, there is power in the cortex at very low frequencies. And here we do the statistics here. So we have 12 scans per condition, okay? And all of the conditions, both sedation, light anesthesia, deep anesthesia, have power in this very slow range compared to postmortem. And then if we increase the frequency range, we find that, okay, this is the typical resting state range. We see also that in light anesthesia and sedation we have power. Okay, so these 2 are good predictors when the resting state, they give signal in the resting state. But then if we go beyond the typical range of frequencies analyzed, not only in humans, because they always filter also in the rat studies, they filter here. And what we find, what I'm seeing here is that there's still signal up to like 0.3 hertz, right? There's still signal in these voxels. And here it really peaks in the cortex. There's a lot of oscillations here, and this is what better differentiates across conditions. These are the p-values, but this is just to show like the Bonferroni threshold is here. And I mean, we're really very, very highly significant differences between conditions. And specifically in this frequency range is where all the 3 conditions, so different states of consciousness or whatever, were maximally differentiated, right? There's nothing in deep anesthesia, there's a bit in light anesthesia, and a lot of power in sedation. So here I'm showing an example for 3 different scans on 3 different rats, but this was consistent in the 12 scans. I looked at all of them. So this is to see when we filter, this is what we see. So I'm not putting any voxels as transparent or anything. This is just the brain mass. But we see that, you see there are some waves there. Now if we look, when we add a bit of isoflurane, the signal is still there, but it's a bit more global and a bit more noisy, right? We still see something, but it's less structured. And then when we add isoflurane at 3%, it almost looks like the dead animals, but we see like from time to time it gets a bit more yellow, then a bit like globally blue, so it's more like a global signal. You see here it's a bit, I will analyze this later to show you a bit more. But so when we, comes a bit of the trick here, we can, to decompose, because what we were seeing here in these first modes here, we see that the oscillations are not globally synchronized. Everything is synchronized together. So what we do is to detect the principal components of the system. And when you look, you have a dynamical system, and if you decompose into principal components, you will see the eigenmodes of the system. You can predict the eigenmodes from the structure using the Laplacian, okay? This is go from structure to function. But if you have already the signals, you just need to do the eigenvectors of the covariance matrix to get the eigenmodes. And this is what you would get at the modes of vibration, let's say in the chamber, or if you get the signals. And so we get here, these are the principal components that we detect with eigenvalue above the postmortem eigenvalue. So it means with energy above a baseline that we will detect. And what we find is that these principal components, and we look at how much they activate in time. So usually in data signal analysis, like in things that have nothing to do with physics, you can decompose into principal components, and you look at when the principal component is active or not, right? You can have it on and off, but it doesn't necessarily need to go positive and negative, and especially doesn't necessarily need to oscillate, right, in a principal component analysis. And what we found here is actually that the principal components oscillate in time. So if we look at the time series of each principal component, so it's how much this pattern evolves over time, it is very, very periodic. Here I'm showing for the 600 seconds of acquisition, but if I would zoom in, you would clearly see here it's going up and down with very sustained periodicity. And this is captured when we do the power spectrum. For example, for this mode, it is oscillating between positive and negative values with very, very always every 4 seconds you would see, 5 seconds you would see the same pattern repeating again. And this is represented by a very narrow peak in the power spectrum. This means that there is resonance. I will show you later. So from this perspective, when we have, imagine, these are the recorded signals. Is it not playing? Sorry, I need the pointer. I want to make it play. How does it work? First, I'm running out of battery, and I have a charger that I put my adapter with. Thanks.
The resident mode is about to run out.
No, but it's very important that we.
Is it gone? Is it all the way in? Is it charging?
Yes.
Good.
I need to put this playing. Okay. Play. Now, put presentation mode. Okay, now it is. Okay. So this would be the recorded activity. Here, there's not, it's just filtered below 0.5 hertz. All the activity, it looks super noisy. It looks a bit like what we see even in humans, but we still see those waves that I was talking about. But it's quite noisy, right? If we decompose into different principal components and we make each one activate according to its temporal signature, right? As I said, it switches between positive and negative values. So here it's positive representation of the pattern that we see also very clearly. If we take all these subcomponents and then we add them up together, okay, we can reconstruct the signal as the superposition of these modes, let's say. And here we see that just with 10 time series and 10 spatial maps, we can reconstruct, we can get back the hidden, like it seems to capture what is organizing the signal in the beginning. It's like, usually PCA is like a denoising technique, so you can always decompose a thing and take only the principal, the components with more eigenvalue and reconstruct it. But here the cool thing is that it is really obeying to this wave superposition principle that you can have different modes of vibration. This is why when you play an instrument, and most of you come from music, right? You can play different notes, and these notes, what you hear, the notes themselves don't interfere together. You hear the superposition of those waves at each instant of time. And then you can decompose it. But also, if you just look at the picture at each instant of time, you don't capture the different modes that are giving origin to it, right? Giving rise to it. So it is one of the things that we can see this as a superposition of waves. then this is like if we look at multiple slices, we try here to do multi-slice acquisition. This would be more or less the slice that we were looking at before, but this is to show that even if we go from front to the back, we did 12 slices, of course, then our TR becomes longer. But to show these patterns are observed across the different slices, and it's clear there is more signal inside the brain, right? Here is still, like we see very interesting patterns in the cerebellum and that, but it's still, there's a lot to explore in that direction. So when we added a bit of isofuran, we found that the modes changed a bit. So we see the same, like phenomenologically, they're the same thing, right? Because there are also these patterns that switch between positive and negative values, but their spatial configuration is slightly different. Especially these last ones looked a bit more noisy. And also the power spectrum becomes broader, so there is less fine-tuned frequency. So it's more like it would do like it changes the frequency over time. So we clearly see here it goes a bit slower, then it accelerates, then it goes slower. But there are these frequencies, so it's still interesting. And then when we go to deep anesthesia, the only mode that survives is the global mode. So we see here in the carpet, so here would be the signals in all brain voxels, 1,500 voxels over time. It's what people call carpet plots. I didn't explain before, but if you see like a vertical line, it means all the signals went up, like to positive, and then all the signals went down to negative in blue. And we see this, but they are more aperiodic, and they don't have really anti-correlations over time. And this relates to things that people have found in disorders of consciousness. That's why I'm interested also. And in coma, you have only these global and slow waves that appearing, so it's interesting. But then what I wanted, then people were arguing, how do you claim this is resonance if you're not predicting the resonance from the structure? Yeah, but we can detect signals. Sometimes we detect a periodic signal from space, I don't know. And if we hear like, okay, maybe there's something. We do the power spectrum of the signal, and we see there's a peak, and then there's a way to see how much a signal, like in engineering, for example, if you're building a thing and then it's vibrating and it's creating a sound like this, you can apply the power spectrum, and then you calculate the resonance Q factor, which is actually how peaked is the power spectrum. And usually resonance is an annoying thing. You want to get rid of it, right, for things to work, or you don't want in a car, if it's doing like, okay, something is wrong, something is not, but it's a way just to analyze the data. So we decided, okay, let's apply the resonance Q factor to the signals associated to each mode. So we have for each mode, in each condition, for all the 12 scans that we did, we can look at the peak frequency across conditions, and we can look at the resonance Q factor. They are a bit both similar, but what was nice is that we find some of the modes have really high resonance Q factor. So that it means that it's how many sustained cycles it remains when it is activated. Imagine there's a mode that remains 15 times, so that's like right, and you bump a ball. And other modes, like the ones in deep anesthesia, it just vanishes, right? Maybe you are from music, so you know even in the piano you can press a button and you just change the damping coefficient. So it here is to show, like, for the same mode, even the global modes, in 3 different conditions, in medetomidine only, it reverberates like 3 times, usually the number of times even in magnetic resonance, and that we take like how many times it rebounds up to around 37% of the original signal, which is 1/E. It's a thing. Also because, as you see here, even in postmortem, the signal can still oscillate below 1/E, but what is important is to count how many times it oscillates before it decays. So then if this is with the autocorrelation function, so it means every time the signal activates, how long, like, it doesn't just decay, it has rebounds, and that is very important to consider, because it means that if you would look in the phase space, again, like we here do the Hilbert transform of the autocorrelation function, we switch it like this, so we don't care about time anymore, but what we want to see is how, when the system is activated, how much it decays back to the fixed point, right? And it decays with an oscillation, right? So it means, like from dynamical systems theory, like the Poincaré diagram of different dynamical systems, this is a spiral sink attractor, which is how the things return back. And there are arguments that everything in nature is a spiral sink. Sometimes some things are just, it's an illusion because they are overdamped, let's say. So when we even do it at the macro scale, this suggests that what we are actually seeing, like when we see these patterns going up and down, if there is this kind of damped oscillatory response, of course this is like a cartoon, in the sense this is what is usually used to represent damped harmonic motion. Wait, I'm not using the pointer anymore. To represent damped harmonic motion is like a spring that goes like boing, boing, and this damping coefficient sometimes depends on the medium. So here imagine you just change the viscosity of the system, you would have more damped system, or as you know as well from music, or even our voice can sound different, or we can play at a different frequency if we just change the concentration of the air or helium in the air. But one thing, so this is also nice, that the same container, so there's sometimes this idea that the container shapes the dynamics. Yes, but the same container, if you change properties of the medium, you can have different modes. So the Laplacian, of course, is a simplification, but this is also because it wouldn't explain why the modes change across conditions, and you can be in a coma, but suddenly you wake up and the system is the same, but something in the system changed very rapidly that makes you become awake. So yeah, overall, just to sum up, the story of the brain melody is that you could have a superposition of standing waves, and these waves, actually these 10 waves were the ones that survived the crosstalk rat, but if I would go into narrow frequencies, sometimes I could detect like 50 modes that in individual rats, and actually there could be an infinity of modes in that level. But they are kind of quantified. Usually people talk about superposition of waves, but it's also quantum superposition principle, in the sense that you can explain everything that you observe, like even a very complex dynamics here. This could be irreversible in a sense. It's like a song. When you play it the other way around, it doesn't sound, but the ingredients composing that sound, at the notes, they themselves as individual components can be reversible, right? If you switch upside down a note like that, the rhythm it generates is the same, or upside down. But when you superpose all of them, it can generate a very complex pattern of activity. And this is what this cartoon represents in a way. Each pattern would be an attractor, like a limit cycle attractor, and then you can go to that attractor for some time, and then you return. But then overall, the sum, like the collective wave function, is the sum of all these different modes. They can or not be interacting. I didn't much explore what is the connection between them, but here, each node or mode here is not one brain region. This is a spatial pattern. This is a collective system. So it's a bit like an orchestra that is playing together. I also tried to make a model of this. So, okay, but here the assumption is, okay, I'm not going to predict them from the structure because of the people have tried and maybe we don't have all the information, but let's assume that the modes that we are detecting, the principal components, are indeed these modes. Now let's make them oscillate, right, from time to time with transient oscillations. And this is where we had an oscillator model. But again, like we were saying, like you can have a stable oscillator. Like you said, you can have a standing wave that is always very constant amplitude. And the Kuramoto oscillators are that. It's just something, it's a stable limit cycle. It's always oscillating, but the amplitude doesn't fluctuate. But then you can have models like the Stewart-Landau, where you can set it in a subcritical regime that it will only oscillate when it receives an input, either a pulse or you perturb it with noise, and it will exhibit oscillations with fluctuating amplitude. And so you can simulate some time series. I make these patterns oscillate at this time series. I fit them to the frequency that they were supposed to be, and I generate like a simulated activity that then if I look at snapshots of it, we see that you can have an infinity of patterns that can be formed from this superposition of these. But then if I do the correlation matrix of these signals, and this goes back to figure 1 that I didn't show here, but in the real signals, if you put a seed here, you have a correlated activity on the other side. And that's the thing, people would assume, and also I worked on that a lot, that these are correlated because there is a structural connection between them. And here the hypothesis is that there doesn't need to be a structural connection as long as they are part of a continuum and they are like two nodes of the same wave, antinodes of the same wave. So yeah, this is the final, again. Okay, but yeah, okay, yeah, it worked. So understanding the mechanisms of functional connectivity, I think, is crucial, because only if we understand, then we can think of strategies to drive them, right? I also think that when we reduce the brain as a network, as I did for a long time, of course, in my PhD and all the time, I was always just working with matrices. We fail, or it may be hiding, you know, essential features, like here the wave patterns that are generated by this superposition of waves is also very interesting, and it seems that there has some properties of fluid dynamics that are not necessarily just blood. So I keep arguing that there is more than just the bolt signal in fMRI signals, the bolt component. And I think these results, in a way, support the hypothesis that has been put forward by Celine and other people, like showing maybe these are standing waves. I, for a long time, had doubts, and now I'm becoming more and more convinced that what we're capturing with LiDAR are actually these, like the attractors. So it's not that they're always there, but with the k-means or these things, we can detect what would be the points that are then driving this complex activity. It's not that you are switching DMN, then somatomotor, then something else. It's just that from time to time, the fluids would resonate, the fluids or whatever it is. Why I said fluids? Now here I want to, so after the discussion, I wanted just to show you things I've been doing. So this is myself. I went to the scanner.
Self-portrait.
So my brain, and this is how would I make it. So this would be the broadband signal that we detected. I just removed the mean from all the signals, okay? And this is my brain. We reached a TR of 68 milliseconds, and it's super noisy, right? But then just to give you an intuition of what the bandpass filtering does. So if I filter here, I do the power spectrum, I see that my heartbeat frequency is around 1.1 hertz. So then I filter in this frequency range, and you see here, even the blood, and you see like some things here. Okay, so this would be what is happening at the heartbeat frequency. Now I will show you if I filter at my breathing frequency. Because even here, the signal doesn't follow the same dynamics, right? And we see here there's a much more here oxygenation of blood. There's also here we see around the brain and inside the brain. So there seems to be a lot of signal here at the level of the sinuses, and this could be either CSF, because CSF is also known to give a lot of signal in the different parts of the brain. And then it's in the typical resting state range. Oh, sorry, because this is important. 1, 2, 3. Okay. At the resting state range, it's very slow. Get ready, because you need to be patient. Stop. It gets very red. And now you see in the front, it's getting more blue, and now it's blue here in the back, and it was red here in the front. And then it goes back. So here it's getting a bit red. But it's very slow. You see, here it's like 55, 57 seconds. I even accelerate it a bit. There's something interesting as well, because here, you see, there's a region here that turned blue, that locks in phase with the signals. Now, okay, now it's red, and then it was blue. Then it's blue, and here the front goes red. And there are papers, there was a science paper showing that the signals in the fourth ventricle, which correlates, that goes in antiphase with the fMRI signal in the brain, which, so some voxels have CSF. They are known because inside the ventricles what you have is CSF. And so how do you call it? Cerebrospinal fluid, because in Portuguese is liquid cephalorraquidiano, it's a different thing. But it's interesting because we need to understand that also inside the brain, it's not just the bold signal activation. Also, it doesn't make sense like when everything turns red or everything turns blue, it doesn't mean that the ratio of hemoglobin really went down across the whole brain, or that the firing of neurons when everything turns red, it doesn't also mean it could, like all the neurons are firing at the same time, because we know that's not happening in the resting state, right? So there are a lot of questions that these things, like now I'm also doing on mice, and here you see like, here they are deeply anesthetized, but you see everything turns red, then everything turns blue, then everything turns red again, then, so it's so macro scale that everything turns red, even in the cortex, that I don't know, but not all the neurons are firing at the same time to drive the bold response everywhere. So there could be things that we're not fully understanding about these signals. And this is at frequencies that are much lower than the heartbeat or breathing, so it's not really a thing. But could this be linked to fundamental modes of resonance in these chambers? Well, finally, what I want to finish is with this sentence by Socrates saying, the more I know, the more I realize I know nothing. And, well, I think we're all just trying to make sense of these things, but really, I don't know.
I mean, obviously I forgot to say about this. She's a real L'Oreal woman of science, and I think we've seen that today. What a tour de force. I was wondering whether that scan was done when you're doing talks as well.
I did holding my breath like for 2 minutes. I still see the breathing rhythm is still there, even when I'm not breathing. Like there are things like, I don't know.
Yeah, people in the ear that can do other things to you. Practicing. But I'm sure there are lots of exciting questions, because I can see already one of those people will now ask you a deep question.
Yes, thank you for the excellent talk. This is a broad question. So you mentioned that you can do this decomposition of stimulation, pretty much any system, right? I mean, so what do you think is different about the human brain compared to just physical systems that are inanimate, seemingly inert, without consciousness, without all these other complex variations?
Okay, of course, then there's the neuronal system. What I'm now hypothesizing, and I really like to make clear, these are hypotheses, but that there may be things modulating like this with these macro scale patterns. They kind of modulate neural activity. They may not be necessarily driven by neural activity. Imagine if they are patterns of resonance of the CSF inside the brain or something, and then you have like an income of positive ions that change the membrane. So they imagine when you are in the DMN, those areas that have more activity in the antinodes, they will change the membrane, so the neurons would be more susceptible to fire, for example, at that level. But then how it relates, so it's a process, and there are many processes in your body that can be replicated in vitro, and that is the thing. I'm not saying this is a unique thing of human consciousness, but this may be a support system for human consciousness. So if you cut this out, imagine this is bringing nutrients to the right place or oxygenation to the right places or something. It is important because it is feeding and clearing also the toxic waste from the brain. And then consciousness, I don't know where it emerges, at what level or whatever, but yeah, the neurons may play an important role. Something also that was put forward very recently now in the last 3 months, they are showing that medetomidine is a glymphatic enhancer, right? So the lymphatics are really it's the system that, how the CSF goes from the ventricles through the brain and then goes out. And it has been shown that many neurological disorders, neurodegeneration is associated with failures in this lymphatic system. It's not that, and this leads to disorders of consciousness, but it's not that the lymphatic is consciousness, but it's supporting consciousness. It's like when you nurture a system, or maybe in any system in a car that is not working well, you have to change its oil, you have to change the things, you have to do the housekeeping, you know. And I suspect that these modes that we are seeing are more associated with the type of housekeeping in the brain. And if it is disrupted, or some flows are not going in the right way, or not oxygenating the right areas at the right time, maybe that can have consequences that lead to disorders of consciousness and things. But yeah, I'm at that level. Then if there is actually a function for these modes, I am unsure. But I feel as well, you know, when you have these things of phase locking, when you have something that goes up and the other goes down, you create a net magnetization. You create the dipole. You have two poles. You have, when everything goes together in the global mode, there are no dipoles. Everything goes together, everything goes down. When you have a dipole, things will flow either from here to here, right? Or you have a directionality. So it could also be driving, you know, the flow of particles to certain areas, like paramagnetic particles can be attracted. This is like crazy speculations, but in the sense that there may be principles that we are not fully grasping, that may be also playing a role. Of course, the neurons are important, but they will also fire depending on the extracellular medium.
So really, it wasn't Socrates you should be quoting, it's Heraclitus, right?
What?
But everything is flow.
Yes.
But then I think there will be a follow-up question here.
Yeah.
In fact, it's sort of related to what you just said, which is emboldened to be a little bit more out there now, which is to what extent do you think the electromagnetic field might be actually influencing? Because that is a kind of a global connected field, and it's been controversial, but you know, then maybe.
I totally agree. Like, so that is our best correlate to consciousness. It's what we use to measure any type of, you know, people in coma or whatever. You put electrodes here to detect the electromagnetic field, you can call it, because if there's an electric component, there's a magnetic component. So indeed, it is there. And indeed, there is a bit of a confusion in the field, because what we learn in textbooks for the signals that we detect in EEG, imagine you have these slow waves of T or collective waves, and they say, okay, for the signal to go up, like you have all the pyramidal neurons firing in phase, like it's really a lot of neurons, even when you're in the resting state, to drive a gamma wave, and they exhibit properties of fluids. So linking to that and to fluid dynamics, there's a thing called magnetohydrodynamics that the guy won the Nobel Prize in the 70s that puts together Maxwell's equations for electromagnetism and Navier-Stokes equations for the fluid dynamics. And what it's showing is that when you have flows of ions, like positive and negative ions, it can generate oscillations in the electromagnetic fields. So the fluid itself, so turbulent flows, can generate turbulent electromagnetic fields. Okay, so this is very interesting, because if you enter in a resonant mode, you've seen like standing waves sometimes in pools, you see that garbage accumulates in some areas of the sea. In the same way, we could see that some particles could accumulate in some. So yeah, maybe doing in vitro something like that that relates, or at least you could create something similar to an EEG signal in a water balloon, because now it's the thing in physics of fluids. There's a paper, 2 papers from last year already, showing how water balloons, they exhibit the same phenomena, you know, of water drop dynamics, like the spherical harmonics of the mini water drop. They go very fast and it's very hard to see. And what they show is that if you have a water balloon, the modes are the same, but the frequencies are much slower, and it still resonates, and it still creates this standing wave pattern. So there are these very nice analogies, but putting together fluid dynamics with electromagnetism that I think could, but yeah, again, how this relates, so the neurons in a way are transistors beta inside this electromagnetic fluid. Let's
Yeah,
say.
the point I was kind of hinting towards is the more controversial point that the EM field actually has downward causation back onto the firing rate of neurons.
The causal consequential, yeah, exactly. But it's hard to put that forward in grand applications or things, but I totally agree. Some people say, oh, that's completely nonsense, but I think that field is growing a bit. What is really causing what? Because there's no direct proof that what we're seeing in rest. Task-driven bold response, yes, there's a causation because it's you know, this, the neurons are firing, they need more energy, they will drive a bold response. But then there are many other components in the signal that are not necessarily driven by that, and they may be modulating the firing of neurons.
Sort of connected to Shannon's question about that unified electromagnetic field. Maybe one way of looking at it would be to measure resonance between different networks. So have you done that to see? I mean, you talk about broadband, but what about the kind of like wireless connectivity? Almost that you have mirror neurons. That's already been kind of studied, but I wonder if what you've done applies to several networks at once.
Okay, so I mean, so here when the net, there can be different things. Either the different networks are really influencing each other, which means that when it's activated, one wave pattern activates, it will drive another. Or if it's a purely wave phenomena and electromagnetic waves and sound pressure waves have this property of superposition, which means they don't interact. This is why we can see things, we can hear things, because the wave that comes from that leaf in that tree far away came to my eye without interfering with the rest, and or in the sound, you can hear all the notes. It doesn't, you can decompose it, right? So there is no interaction. It's a superposition. It's like this multiplex division of things. I don't know. And then somehow we would have something that interprets and puts everything together.
Yeah.
Or indeed
different sort of neural networks.
Yeah.
You see brain activation that kind of mirrors.
From one.
Yeah.
Okay. I mean, I follow the kind of Resonance Foundation. I don't know if you know this guy. Well, they already cited my work. I was super happy to see that because I've been following them for 3 years already. But of course, they claim that indeed everything is connected together, and that, of course, even I was seeing the other day about. So we are a water balloon because we know we are 90% of water, and somehow this water is connected here. But you know how they kind of detect cosmic rays? I was seeing a documentary the other day. They really put in a field different boxes of water, and then they have sensors at the water to see, because water is very sensitive even to any type of interaction. It really vibrates a lot, like you put a bucket here and it will sense different types of things. So it could be that there are some interactions that we feel.
Like the water balloon theory of consciousness. I think we are very close to having to have a bit of power to further develop that idea. But I think
I will do that experiment. I will do it. I want to put it in the scanner. But of course, it's with CSF. So CSF has also very particular properties because it has like a pH balancer. It is a transporter of the liquid itself. And so I just ordered 100 milliliters of the CSF, 500 euros. And actually, but to have like real CSF is very hard to get. Now I ask for a budget. 1 milliliter of CSF costs like from a rat. Costs 3,000 euro, and it's frozen. And then there are papers showing that after 1 hour after that, the CSF already changes properties. So there can be properties in this particular fluid, and there are fluids that have very particular, like magnetohydrodynamics applies to any salted water. So it's not just water. H2O doesn't have, you need at least positive and negative ions inside.
So it's like saliva, really.
It's also like in those sites where they sell the biofluids, you can buy not only saliva but also things. But yeah, in the end,
Dark web, don't try it. Don't try it. But we'll leave that for the moment. We'll come back to the dark web
There's a
The
question here,
disremark
question there.
of disorders of consciousness is the CSF
Let
problem.
the people who know things ask first.
My first specific question. So in the, I believe in the discussion slide, you said that the resting state networks would be antinodes of the standing waves. And I was just curious. how
Yes,
that is-
exactly. So in the wave theory, it's different from network theory, where you have the nodes and the edges, right? That's network. In wave, the nodes are the points of no motion. It's not like the nodes in a network. So if I put here, sorry, I think I was going to write this, but then I kind of decided to go to sleep. It's actually, and I was putting this arrow here, but then I didn't continue. Yeah, so imagine in the first one, there's one antinode, which is the center, and the boundaries are the nodes. The points of no motion are the nodes. In the Chladni plates where the sand accumulates in the points of no motion, in the white areas here. Okay? So it accumulates in the white parts there. And because in the others, actually the plate is vibrating a lot, so the sand is moved away and it finds the places. So it could also be like, and some particles are known to accumulate in the points of no motion, but in the points of high energy, you can have like attraction of paramagnetic particles, for example, you can put there, because they are attracted to the high field.
So, final question.
Just my since what I did catch on to was the water loop consciousness. I think that's my level here. But I was wondering with the what music are you playing? Is there because this is first, no? So it's a signal, I'm guessing, but for humans, you're playing music, no? Or no?
Okay, this is just resting state, and when I say hertz, is that so? This would be so. There's no music. No, there was a study.
The brain is working itself.
Yeah.
Exactly. The brain itself is making music, and we are detecting these waves at ultra slow frequencies. All the frequencies we detect from the brain at high power are below the hearing threshold.
Okay.
Okay.
Yeah.
And these are real. So the hearing threshold would be around 20 hertz or something. But still, EEG waves are electromagnetic. We cannot hear them. We would need an something to amplify. I don't know if these are linked to pressure waves because it's what you see here, for example, and this could give rise to changes in the fMRI signal because you could have more proton density, less proton density, more proton density, you have like waves of things. It looks like, of course, we see the structures like, and we have ventricles and things, so they could be indeed some small vibrations happening there, but I don't know if they are pressure waves. So there are three types of waves. There are chemical oscillations, pressure, or electromagnetic. I mean, I'm still confused about what exactly we're getting.
Yeah. Or maybe clarification.
Actually, can I have
Yeah.
Okay, so this is a really, this is amazing work. This is super exciting. I just wanted to understand about how do we interpret this decomposition into waves. Obviously, any signal can be decomposed. The thing is, if it's a random signal, the decomposition is going to be like the power spectrum is going to be very flat or something.
Yeah,
So
all of
the
them
question is
that's what we do in the postmortem scan.
Right. So you got this kind of maximum, a random signal gives you this huge entropy. So it's not interesting.
And all of them have an associated eigenvalue, right? Each of them has. And then you take, in the postmortem, I take the one with the highest eigenvalue, okay, which is the mode with the strongest eigenvalue in the dead animal, okay, and that is my baseline to detect the others. So which are how many modes we have that are detected with energy that disappears after the animal is dead, right? So we can really find a threshold there. And then another thing that proves, I do to one rat, I do the eigenmodes. I do to another rat, I have slides that I have a lot of studies on it, I get the eigenmodes. And then they are the same. You look at them visually, that's why I insist on plotting the videos and everything, because it's not just a random decomposition. If you decompose noise in 10 modes, you take the first 10 modes, these things of what is the ones that represent 95% of the variance. You can always get a number of modes that it doesn't mean that those modes are meaningful or not. That's why the postmortem scan was valuable to define what is the baseline. And at the same time, they were very consistent across animals, of course. And we detect similar modes, especially in humans, and they are very consistent across humans. So if it was just noise, would we see this? But they seem to be highly constrained by the structure. You see, like they peak in power within this kind of anatomical boundaries. If we look at the anatomical slice, we see that there are boundaries. And one property of resonance, it's like a thing, it needs a boundary. It needs to hit the wall and come back somewhere, right? And go like time. If you don't have a boundary, it just goes on forever.
So my question is like, how do we know what is the actual useful information, the complexity that matters for brain, rather than just the noise? Is there some way from this can we estimate what part of the entropy is actually useful in representation?
Yes, okay. Entropy would be a more coarse measure because it doesn't really go into the detail of which specific pattern is changing between conditions. That's why all the different papers I'm doing, even with LIDE, okay, we now have autism, 1,000 subjects, 1,000 controls, and they significantly have less of the default mode network, all of them, and it's there with highly significant. We did it with depression recently, another data set. One mode is differentiating between conditions with 1,000. So and it's one mode, it's not like, so there's a common thing. And I think that's when you define, is this meaningful or not? Because if I was just looking at healthy ones, then you don't know if they are relevant or not, right? You need two things to compare.
I think it's time for those water balloons. So I think, can we thank you again, Joana?